Hello, Dr. Eisenstadt. My name is Eleni. I am the CEO of the Cure Starts Now Australia, and I am here to announce with my colleagues that we are awarding you a partial grant of $156,914 for harnessing developmental neurobiology to reprogram the cell origin of DMG. Congratulations. We are so proud to be able to do this for you and to support your valuable research. I'd now like to hand over to Tamara, who has a few words that she would like to say.
Hello, Dr. Eisenstadt. I'm Tamara Ikas. I'm with the Reflections of Grace Foundation, and I'm representing the 27-member DIPG DMG Collaborative joint participation here in this grant. We are so proud to be able to award this to you and be able to fund this for you. You know, we are obviously a group of foundations that formed by moms and dads and relatives that have, wanted to find something that is a better option and change the needle for children that are diagnosed with DIPG and DMG. So we are so pleased to have this grant for you, and we are so pleased that you are researching such a critical need in our community. I know Keith wants to say a few things, so I'll hand it off to him.
Thank you, Tamara.
Dr. Eisenstadt, pleasure to talk with you. Thank you for giving us some time here to talk a little bit about your grant, but also an award for it. We've met, obviously. I'm with the Cure Starts Now, and I represent the 52 chapter organizations of the Cure Starts Now that are behind this and are very supportive to see your research go moving forward on it. And speaking of that, I think it's all about also profiling what this research is so that we can help to get the word out there and to better understand it. And in looking at your grant, this whole concept of reprogramming the DMG cell of origin with the help of DLX2 is kind of a novel concept. Can you kind of talk a little bit about this and what makes this special?
Sure. It might help if you put up the slide just because I'm going to use some words that people may not be familiar with.
Certainly.
And I'll just first briefly mention a little bit about myself so you can appreciate my journey to get here. I'm a pediatric neuro-oncologist. I was one of the first trained who then returned to Canada, and I'm now in Australia for the last 4 years at the Royal Children's Hospital, where I direct the Cancer Center. When I went in to do my lab research in the 90s, I started off in a molecular neuro-oncology laboratory, but I really believed back then, and I had no basis for it other than as a pediatrician, I wondered why couldn't some pediatric brain tumors be disorders of normal development? We just didn't have the tools, we didn't have the knowledge, we didn't even know what questions to ask. And so when I came back to Canada, I set up my research lab. Essentially, I decided to focus entirely on brain development, and clinically, I do clinical trials. And I have to say, and I know I'm preaching to the converted here, that every child I saw with a diffuse midline glioma or DIPG, as we called it back then, died, and they still do, including after CAR T cell therapy earlier this week in Melbourne. And so I was waiting to have some biological connection between brain development and brain tumors. In other words, between my two day jobs. So as it happened, this transcription factor that I've been studying since my postdoctoral training at UCSF with Professor John Rubenstein worked on a homeobox gene called DLX2. And the details aren't that important other than this is a transcription factor, and one can think of transcription factors as proteins that bind to DNA of thousands of other genes, and it can either turn thousands of genes on or thousands of genes off. And so my lab's been really trying to figure out how that works. And as it turned out, we found out that DLX binds to the gene promoters of enzymes called GAD1 and GAD2. And although that may not be that relevant, you'll see in a moment how it might be. GAD1 and GAD2 are genes that convert glutamate, which is an excitatory neurotransmitter in the brain, to GABA, which is an inhibitory interneuron, basically shuts things down, the synapse potentials. And so, as it turned out, recent studies, not from my lab, but from others, have shown that the likely cell of origin of diffuse hemispheric glioma, these are the pediatric high-grade gliomas that are more in the cortex of the brain. Those are the cell of origin is likely a GABAergic interneuron, and I've been a collaborator from the groups in Boston and Stanford that published that work in both 2020 and 2024. At the same time, my lab has been showing that in the cells that are programmed to become GABAergic, express this inhibitory interneuron, it shuts off the entire program of oligodendrocytes. And the oligodendroglial progenitor cell, or OPC, is now thought to be the cell of origin of diffuse midline glioma. And there's several studies, majority coming from Mariella Philbin's lab and others that substantiate that. So here I am, a pediatric neuro-oncologist whose lab is really focusing on a transcription factor up until recently relatively obscure that controls the cell fate decision between becoming a GABAergic interneuron and shutting off the entire OPC program. So when I moved to Melbourne, and the reason for that was to have access to advanced genomic platform, we've been able to get the ball running with advanced omics platforms, including single-cell RNA sequencing technologies, ATAC-seq, multi-omics, and spatial transcriptomics. And we've more or less confirmed that this hypothesis is correct. Furthermore, when we took Oren Becker's mouse model, it's a genetically engineered mouse model that gets diffuse midline glioma, or DIPG, in the brainstem, and got those cells and put DLX2 in, we could actually turn on GABAergic fate and shut off OPC fate and restore what's called histone trimethylation of lysine 27. Next slide, please. So looking at the schematic, when you have this would be in the normal brain, a neural stem cell can really become one of three types of cells: an astrocyte, top, middle, in purple, an oligodendrocyte. These are the cells that make myelin for the central nervous system in green, and an interneuron. And these are the neurons that express GABA. And the cell of origin of diffuse midline glioma is an OPC, or oligodendroglial progenitor cell, and the cell of origin of diffuse hemispheric glioma is a GABAergic interneuron. And DLX controls the fate switch. In other words, it promotes GABA and shuts off OPC. Hence, one could then suggest that if we could overexpress DLX2 in diffuse midline glioma, we could really reprogram those cells to become more like GABAergic interneurons. At the same time, in diffuse hemispheric glioma, we can knock down DLX2 and we can make them less aggressive. And we have some very good in vitro data. So next slide, please. So what does this all mean if you were trying to make an elevator pitch for pharma? And so I've been thinking about this for a long time. I think if we're going to take 0% or 1% long-term survival for DMG and make it, in my lifetime, 20 to 50%, we're going to have to follow the neuroblastoma model. The neuroblastoma means it's not one size fits all, and it's not one drug. It's multi-dimensional therapy. So you need local control. In this case, biopsy is now becoming the new standard care to establish the diagnosis, radiotherapy for local control, and there may be some really cool things that others can do for blood-brain barrier disruption or convection-enhanced delivery. That's to really improve radiation's effect. The next step would be immunotherapy. And as you know, CAR T-cell therapy is showing tremendous promise, but there are others working on cancer vaccines, immune checkpoint inhibitors, whatever. And this, in some ways, if I would use the analogy of a tree, would essentially knock down the tumor, but you're not getting to the roots. Whereas differentiation therapy, which are neuroblastoma or retinoids, we don't have good differentiation therapy for glial tumors, including DMG. We really need to bring in this concept of epigenetic developmental reprogramming. So whether it's my lab or others who take our lead, we're working towards figuring out the molecular mechanism, how to really augment the effects of DLX, and it may be that there are signaling pathways downstream or upstream that can turn DLX on or off and essentially reprogram the cell of origin of DMG. So that's the basis for our entire research program.
Well, and obviously going through it, it's a very encompassing program that you put together with it. And we see that this strategy can be utilized in this multimodality strategy, which, certainly is something that we're very interested in. So we wish to offer you thanks and congratulations on this. And certainly we look forward to learning more.
Well, thank you again. This is really unexpected and happy news for my lab, and we're delighted to become part of your extended, I don't know if you can call it family of researchers. But I think I've learned over the years that we need to work together because this is a very tough fight.